Good morning, everyone. My name is Geneviève de Blois. Welcome to this month seminar series. It's a pleasure today to have Dr. Claudia Kleinman and her trainee Selin Jessa for this monthly seminar. Selin Jessa will start the seminar. She's a computational biologist who finished her Ph.D. at the lab of Dr. Claudia Kleinman. She's finishing her Ph.D. In Quantitative Life Science program at McGill University in Montreal. She completed her undergrad training in computer science and biology at McGill University also. Her doctoral work is focusing on using data-driven approaches to understand the origins of pediatric brain tumors by studying relevant regions and cell types in the normal developing brain. So the title of our seminar today is Pediatric Glioma with K27M mutation in canonical and non-canonical histone tree have distinct origins. So Selin, the floor is yours.
Perfect, thank you. All right, so I'm really excited to be sharing some of our recent work today on understanding the origins of H3K27M mutant gliomas, which is one of the focuses in our lab. And so one of the very puzzling things about these tumors is that the same K27M mutation occurs in multiple variants of histone 3, but the resulting tumors have some different properties. So histone 3.1 and more rarely 3.2 K27M occurs exclusively in the pons and usually with activating mutations in ACVR1, which is a BMP receptor. And in contrast, histone 3.3 K27M occurs both in the pons and the thalamus and with distinct partner mutations. And so one of the driving questions has been, why do these patterns of histone 3 mutations associate with different tumor types and brain regions? Now, histone 3.1 and 3.3 are different variants of histone 3, and histone 3.1 in particular is coupled to the cell cycle, so it's only incorporated into the genome during S phase, whereas histone 3.3 is cell cycle independent. And these two histone 3 variants also have distinct deposition pathways and patterns of localization in the genome. So the differences that we see in the tumors could be due to these distinct functional properties of histone 3.1 versus histone 3.3, but it could also be due to their differences in partner mutations, differences in the brain regions where the tumors arise, or different cell of origin. And so in this project, we were trying to distinguish between these different possibilities. And so we assembled a large cohort of both patient tumors and patient-derived cell lines, which we profiled by single-cell RNA-seq, bulk RNA-seq, and ChIP-seq for different histone marks. Now, the consensus on the cell of origin for histone 3K27M gliomas is in oligodendrocyte precursor cells, which give rise to oligodendrocytes, and these are the myelinating cells of the nervous system. And importantly, the OPCs persist postnatally, and they have some self-renewing and differentiation and regenerative capacity. So the first thing we asked in our single-cell RNA-seq data was, what do these tumors resemble in terms of normal cell types? So in these figures, every bar in the plot represents one patient sample, and we're looking at the breakdown of the cell types we see among malignant cells in each sample. And what we found is that both H3.1 and H3.3 tumors predominantly resemble the oligodendrocyte lineage, usually transcriptionally resembling OPCs and with smaller populations of astrocytes, oligodendrocytes, and cycling cells. And so at the single cell level, these tumors transcriptionally resemble OPCs, which is consistent with the hypothesized cell of origin. So next we honed in on the major differences between the tumor types by comparing 3 different types of molecular data. And so in this figure, every point represents one gene. The x-axis is the differential promoter histone 3K27 acetylation between H3.1 and H3.3 tumors. The y-axis is the differential trimethylation, and the point color represents the RNA-seq log fold change. And so what we find is that when we compare H3.1 and H3.3 tumors across these data types, the major differences are limited, first of all, but second of all, they occur at patterning genes, and in particular at 2 transcription factors, NKX6-1 and PAX3. And this dichotomy in NKX6-1 and PAX3 activation was pretty consistent across data types. So in H3.1 tumors based on ChIP-seq, these samples had K27 acetylation at the NKX6-1 promoter, as well as chromatin accessibility. And they also had K27 trimethylation at the PAX3 promoter. By bulk RNA-seq, we could see that H3.1 tumors always activated NKX6.1 and rarely PAX3, whereas the H3.3 tumors were a little bit more variable. And then by single cell RNA-seq, we could really zoom in on the different cell type populations we see among malignant cells. And in that case, the expression patterns were pretty much binary. So NKX6.1 was mainly detected in H3.1 tumors and PAX3 in H3.3. And then we also confirmed this at the protein level. So by immunohistochemistry, H3.1 tumors activated NKX6.1 protein, both in patient tumors, but also in xenografts derived from patient-derived cell lines. And in contrast, we didn't see NKX6.1 protein in H3.3 K27M gliomas or other gliomas that we looked at. So as I mentioned at the beginning, K27M gliomas are postulated to arise in oligodendrocyte precursor cells. And these cells are defined very specifically prenatally in specific waves that are well characterized during development. So in the neural tube, we have patterning set up by two opposing morphogen signaling gradients along the dorsal ventral axis, BMPs dorsally and sonic hedgehog ventrally. And these morphogen gradients in turn activate specific transcription factors in the hindbrain, which then through cross-repressive interactions demarcate these lineage-restricted progenitor domains. And I'll point out that NKX6.1 in particular is very restricted ventrally in the hindbrain. And then in terms of the waves of OPCs, it's known that there's a first early wave of OPC development ventrally from the motor neuron progenitor domain, and then a second dorsally derived wave later on. So the activation of NKX6.1 in H3.1 tumors, as well as their resemblance to oligodendrocyte precursor cells, suggests that their cell of origin might be specified in this early ventral wave of OPC development. Now, because these genes are quite well characterized in the developmental literature, their dichotomous expression patterns are well understood. And so we tried to really extend our knowledge of these two genes later on during development. And so for this, we inferred the gene regulatory networks of NKX6.1 and PAX3 using a single cell RNA-seq atlas of mouse hindbrain development between embryonic day 10 and postnatal day 6. And so in this plot, every gene represents one inferred target of NKX6.1, and they're plotted a little bit like a clock. So they're plot in order of the first time point at which they appear as a target of NKX6.1. And so these are the targets of NKX6.1, and here are the targets of PAX3. And I really want to highlight three takeaways from this analysis. The first is that the dorsal PAX3 and the ventral NKX6.1 progenitors have distinct regulatory networks. So their targets are largely non-overlapping. They only share a handful of genes based on this analysis. The second point is that the NKX6.1 network includes multiple sonic hedgehog and notch pathway genes, but not really BMP genes. And the third is that these two transcription factors and also then the progenitors that they mark seem to maintain their pathway dependencies throughout development because our analysis included samples all the way up to postnatal day 6. So to summarize what I've shown today, H3.1 and H3.3 K27M gliomas both mirror the oligodendrocyte lineage transcriptionally based on single cell RNA-seq analysis. But the major differences we see between these tumors across different data types are at patterning genes. And in particular, H3.1 K27M gliomas activate a transcription factor called NKX6.1. And this suggests that they might be specified during an early ventral wave of sonic hedgehog dependent oligodendrocyte precursor cells. And so altogether, this constitutes molecular evidence that these H3.1 tumors might have an anatomically distinct origin from other K27M gliomas. And with that, I want to thank my supervisor, Dr. Claudia Kleinman, and all the members of our team who were involved in this project. We worked on this project quite closely with the team of Dr. Nada Jubato, and in particular, Shakur and Ashat are my co-authors on this study. And want to acknowledge our collaborators and funding sources, and I'll be happy to take any questions. Thank you.
Thank you very much, Selin. I would like to invite everyone to ask questions. If you can write it in the chat or raise your hand. I'm not sure I can see the questions. Okay, I have some. There's a question by Alan Underhill, who's asking, how are you assessing target genes to PAX3?
Yeah, thanks for the question. So the method we use is a framework developed in Sten Ert's lab called scenic for single-cell RNA-seq. And the way this method works is it looks for sets of genes that are co-expressed with transcription factors in single-cell RNA-seq data to get an initial set of possible targets. And then it does some pruning by restricting those possible targets to only genes which have motifs for the given transcription factor in the promoter regions of those genes. So by that, we get this inferred set of targets for each transcription factor based on the specific samples that we include in the single-cell RNA-seq analysis.
Okay, thank you very much. I don't see any other question right now. Oh, yes, there's one. Okay. He's saying, thank you. Do PAX3 motifs have a specific architecture?
That's a great question that I am not sure. One of the limitations of this analysis is that for transcription factor families with similar motifs, so like the PAX family in general, there's a harder time distinguishing between the different transcription factors, and I'm not sure the specificities for PAX3. But PAX3 does have to be expressed in those cells, so from that we can get confidence that the inferences are more related to PAX3 expression specifically.
Thank you very much, Selin. I have a question regarding, maybe this is something that that Dr. Kleinman will address, but what's the signal that drives this switch or this this specificity between each transcription factor?
In the different tumor types?
Yeah.
I think I'll leave that one for Claudia.
Okay. There's another question by Ethan Hollingsworth, who says, great talk. Have you done any in vitro work with these cell lines, knocking down these two transcription factors?
Yeah, that's a really interesting question. We haven't done any in vitro work knocking down these transcription factors specifically, but Claudia will talk about some of the in vitro work exploring why we might see this origin. Okay.
Okay, thank you very much, Selin.
Thank you.
I don't see any other question for the moment, so we will now go to our keynote speaker, Dr. Claudia Kleinman. So it's a great pleasure to welcome Dr. Kleinman today for this seminar. Dr. Kleinman is an associate professor at the Department of Human Genetics at the Faculty of Medicine in McGill University in Montreal. She's also a full-time investigator at the Lady Davis Institute for Medical Research and associate member of the McGill Center for Translational Research in Cancer and a researcher at the Lundmer Center for Neuroinformatics and Mental Health. Her research exploits genome-wide technologies and data science to understand mechanisms of gene expression. She has interdisciplinary training that combines molecular biology, computer science, statistics, and evolutionary biology, which she applies to study pathological, transcriptional, and RNA processing events, focusing particularly on pediatric brain tumors. Dr. Kleinman has received numerous awards through her career, including from the NSERC, FRQS, CIHR, and she currently holds a career award from the Fonds de Recherche du Québec Santé. She's also a recipient of the Bernard and Francine Dorval Prize for her work on mapping the development of human brain at the single cell level to define origins of pediatric brain tumors. So thank you very much, Dr. Kleinman, for accepting to present your work on today's seminar. So the title of your talk is Identity Matters, Pathogenic Mutations in Context. We're looking forward to hear your presentation.
Thank you so much, Genevieve, for the invitation and for the kind introduction. And thanks, Selin, for the presentation. I'm very proud of all your work that you're finalizing in the lab. So I'll step a little bit back from Selin's talk and give more context and broader context of what we're doing in the lab, which is trying to understand the effect of the mutations in the right context. And I hope I will convince you that identity matters for many reasons, but here at the molecular level. So one of the driving questions in the lab that we have, and it's always puzzling, is why do we have these ubiquitous genes? These genes expressed everywhere. So, but the mutations produce a very, very restricted phenotype. So at some time in the lab, we worked on the RNA-3, for instance, that translates transcribes tRNAs, ribosomal RNAs, which are needed and required in every single cell. But then specific mutations in this context will give very, very, very defined phenotypes. The same goes for many brain diseases, and particularly ones that are caused by epigenome-related genes. So we have the Rett syndrome, the Sotos syndrome, caused by genes that are expressed and needed in every single cell. But alterations will give you very, very fine phenotype. So this is not anecdotal. If we look at the 1,000 or something Mendelian disease characterized, we'll see that most of them will actually be affecting one or two tissues at the most, but the expression pattern of these genes follows the exact same pattern of all other genes, which is expressed in many tissues almost, and very often, very ubiquitously. So the case study in the lab for this question right now is brain tumors, these tumors that we know are caused often by epigenetic modified epigenetic related genes and developmental pathways, and that have the same type of pattern where the driver genes are ubiquitously expressed, affecting the brain, in this case by brain tumors, and sometimes even lowly expressed in the brain. So this is data from the GTEx report. So you heard from Selin, and that will be the focus on the talk today, is from these brain tumors, we'll be focusing on the Histone one, the Histone-driven ones that can, mutations can hit either Histone 3.1, 2, or 3, different Histone variants, and two positions, glycine 27 or glycine 34, and depending on the specific mutation and the specific variant, we will have a very specific and very different clinical manifestation in terms of survival, but also in terms of this anatomical location and an age pattern. So you heard from Selin, H3.1 will hit mainly the pons and mainly driven by partner mutation in ACVR1, whereas H3.3 will occur throughout the brain with K27M hitting the midline and the spinal cord, and G34 mainly the cortex. And each variant and location associated with age, but also with a set of very specific partner mutation. So a question that is puzzling me for years now is, why do we see this specific combination in patients? What makes some contexts more permissive, tolerant, or sensitive to these mutations, so that we see them more often in patients? And the model that we are working under, which we are working right now, is a model of impaired development and differentiation. So to produce the hundreds or thousands of cell types that we have in the brain, during normal development and differentiation, we will see this stepwise massive reconfiguration of the epigenome as the cells differentiate and commit into cell types. And this is quite concerted and regulated. And when we have a histone mutation in one of these progenitors, this process is impaired and the cells become frozen, probably in time, in a state that is quite oncogenic permissive. If you think about it, these early progenitors are quite close to a cancer cell in the sense that they self-renew, they proliferate, they migrate. And so all they need afterwards is a second hit to produce a full-blown transformation. And one important part of the model that we, under which we're working in, is that different lineages and different progenitors will give rise to different tumor types. So the first question in the lab is this model true? Or is actually the different clinical manifestation a product of the oncogenic reprogramming after these very, very deleterious mutation in the Histone gene and other epigenome genes? So the strategy, as you heard from Selin lately in the lab, is to try to understand in quite detail and a high resolution, preferably at the single cell level, the normal development of the brain lineages, and then use this reference, this as a blueprint, to interpret our tumor profiles. And so a couple of years ago, and I will spend not so much time on this because it has been published, we reported the results for the G34 tumors. So you heard from Selin that the consensus in the field for the K27M tumors is an oligodendrocyte progenitor cell. What we found when looking into the G34 tumors is that they don't mirror an oligodendroglial lineage, but they actually mirror an interneuron lineage, and that the second hit is predominantly on the oncogene PDGFRA. So this was a work in collaboration, as usual, with my partner in crime, Nada, and the work of Selin, Carol, Shria, and Jihad, very talented people that are Carol and Selin are flying away from our labs, and we're crying about it. So what we found in this particular tumor is that mapping to the reference, developmental reference, they really mirror an early interneuron lineage. They are depleted of signatures from mature neurons, OPCs, and glial lineage. And the second finding, this was by Carol Chen, is that this partner mutation, predominant one, is mainly in the mutations in the extracellular domain of PDGFRA. And when this tumor is not mutated, it is even still overexpressed here. So back to our question, why are we seeing this particular association of mutations in these tumors? In this case, it's what quite beautifully explained by the hierarchy of transcription factors that are actively involved in committing cells into the interneuron fate. So cortical interneurons are very well described to be originated in a progenitor niche that is transient in development in the ganglionic eminences and governed by a triad of transcription factors, GSX2 and DLX1 and 2. And the epigenome and transcriptomic profiles of the tumors mirror and match this hierarchy of transcription factors with activation of these, again, causal to fate commitment genes. And what happens is that these transcription factors are next to the oncogene, which provides an opportunity specific to this lineage to co-opt this oncogenic pathway. So G34 tumors will activate not only PDGFRA and GSX2, but also regions upstream of GSX2 that have been experimentally shown to be enhancer of GSX2, and so active exclusively in this region and these progenitor niches. So the tumors show an epigenome profile, and including the 3D conformation, where the oncogene is making close contact with these active enhancers, specifically in the lineage of origin. And so that provides an opportunity for co-option of an oncogene that would be explained by a specificity of this chromatin conformation in one specific lineage that would link the two pieces of evidence together. So that's what's happening when we went into the normal brain and profile the epigenome of this region. Let me walk you through these data step by step quite quickly because it's published. So in the embryonic stem cell state, PDGFRA is not making any contact with GSX2 or the enhancers. In the dorsal regions of the brain where interneurons are not inborn or generated, we still don't have any link, any contact, 3D contact with this active enhancer, because it's specifically in the ganglionic eminences in the region of the interneuron origin where we have this chromatin conformation that puts together PDGFRA and oncogene and an active transcription factor. So in this case, the oncogene is not normally expressed because it's marked by K27ME3 and the epigenome conformation is not reflective of an active gene. But we just need a tiny little alteration of this epigenome conformation to produce a tumor. So in this case, what we're saying is that the origin of these tumors is restricted to a specific domain, and the explanation for that is an epigenome conformation that is specific to this domain that will favor the acquisition of an oncogene that is very close by. So that was the case for G34. If we go back to the rest of the gliomas, so the K27M, that we have them occurring through the midline and the spinal cord, and we have a set of different partner mutations, etc. Again, in this case, the cell of origin is postulated to be in an OPC. So we can go back to the same question that we had before. We have here a similar model where different mutations and different histone alterations and different tumor locations will actually correlate with different lineages and different anatomical progenitor niches. Or are we having reprogramming of the epigenome based on the mutations? This could be very well the case because the different histone variants have different deposition along the genome, different relationship with the cell cycle, as Selin was explaining in detail, different chaperones, so different contexts of action. Also, they produce a massive dysregulation of the epigenome with a drastic depletion of the K27ME3 mark, some downstream dysregulation of other marks like K27 acetylation. And they have been postulated to be reprogramming the enhancer landscape. So the different clinical manifestation of the tumors could be very well due to this effect of the mutations. So the questions we try to ask is here, can we uncouple all these three effects? The cell intrinsic properties of the cell of origin, are they different? And why those cell intrinsic properties are favoring one or other tumor type? The downstream effect, oncogenic effect of the mutations. And the third one is the extrinsic, cell extrinsic effect of the regional and developmental timing where these mutations occur. So we have the environment interactions and interactions with other cell types. So coming back to the work that Selin introduced today, we profile these tumors. And I want to just remind you the type of tumors that we have. So we have H3.1 here, H3.3, we use G34 as control. And we also include in this cohort PFA ependymoma driven by ESIP that we use as comparators because ESIP is a K27M mimic. So in this tumor entity here, we will have a similar epigenome effects at some level, at the level of K27M3, but in a completely different cellular context. So that allows us to ask the question of the effects of the different mutations in different contexts, quite different. And because we also have one patient that is a rare occurrence of an PFA ependymoma driven by H3.1 and not by ESIP. So we have this very, very rare and interesting patient. So now that we have the numbers, we can start asking the question with the analysis that Selin was describing as tumors in the same location, but hit by different mutations, or tumors in different locations hit by the same mutations. What do they have in difference? And so what we see is a very clear, in both cases, dysregulation of the Hox clusters. So the Hox clusters often appear as differentially expressed genes or differentially dysregulated genes in these cancer studies. And sometimes they are actually the result of an oncogenic aberrant expression of these key transcription factors that will have massive effects downstream. But when we see so many, we ask the question, are they actually being an oncogenic consequence of these mutations, or are they actually reflecting a fossil reminding signal of the cell of origin? So this audience is very familiar with Hox clusters. I will just remind you a few key points. So these clusters will be regulated by the very beautiful mechanisms of collinearity that roughly correspond to the anatomical position where the body plan is being specified. So in the anterior posterior axis, when specifying the nervous system, we will have all the Hox clusters silent in the telencephalon, and then in the hindbrain and the spinal cord along this axis, we will have them partitioned in two subdomains that where the boundaries shift and being positioned very precisely along the body plan and regulated by a series of very concerted, redundant, and evolutionary very conserved layers of regulation, so that we ensure that each one of the Hox genes in each one of the clusters is expressed at the right time in the right place. So we have an active subdomain and an inactive subdomain marked by K27ME3 and then with knots tied with C6CF loop. So very, very structured epigenome structure here. So the question is here, what happens in the tumor? Are they having an abnormal oncogenic expression, or are they structured? And we find this beautifully maintained structure in these clusters that roughly correspond to the anatomical location of the tumor. So the thalamic tumors that should have the Hox genes silent, they have the bipartite domain, but silencing the whole cluster with K27ME3 despite the lack of K27ME3 that we see genome-wide. And the hindbrain and posterior fossa tumors, again, bipartite domain with an active subdomain and an inactive one, really beautifully marked by K27ME3 and PRC2. So maintaining this structure here, this is Hox-D, but it's the case for all the Hox clusters in each tumor entity. Again, very well structured, silent in the thalamus, and the other tumor entities showing bipartite domain, quite well organized and with subtle differences between the tumor types. So in the thalamus, we see them silenced completely, but there's another set of transcription factors patterning genes that are active there that are equally telling about the origin. So these transcription factors are involved in the segmentation, this time in this diencephalon, and the configuration, epigenome configuration and expression configuration of these genes point to a very specific protomer here that will give rise to the thalamus proper, as opposed to the P1 that will give rising to the pre-thalamus. And again, K27ME3 here beautifully deposited. So what this data is telling us is that each one of these tumor entities is actually originating in anatomically distinct progenitor niches. And that even the tumors that are in the same location have subtle differences. And here I have a typo that I forgot to correct, where if we look at Hox-D8, for instance, we see it active in the K27ME3. And this is what we see here. It's not genomic noise, but it's actually switching from an active domain to an inactive domain in the other tumor type here. So it's quite well defined and quite beautifully marked. So Selin showed you these results. I will go very, very quickly. If we want to narrow down the differences between H3.1 and H3.3, we can find these opposing patterns between PAX3 and NKX6-1 that is consistent across analysis, whether we look at promoter regions, we look at genome-wide acetylation, we reconstruct with Stephen Mack at St. Jude gene regulatory network space and super enhancer analysis. We always find these opposing patterns of PAX3 and NKX6-1, and that is also consistent across data modalities. So clearly in independent samples obtained by the Dana-Farber Institute, after the fact, what we see is that H3.1 gliomas arrive from this very defined progenitor origin in the ventral developing brain. So that's where the cells are specified. So that's what Selin has been showing. So I want to go back to the vulnerabilities here and why do we care and how this is explaining some mutational patterns that we see in the patient. So this opposing morphogen gradients from Notch and BMPs is, again, evolutionary conserved across different systems in the body plan specification. It's preserved in the stem cell, in the muscle stem cell specification. To ventral will be NKX6-1 and dorsal will be defined by BMPs. And the mechanisms of why this is so robust and how has been evolved to be so robust and conserved are quite well described since, I don't know, 20 years ago. BMP, when these regulatory networks are intrinsic to the specification of the cells and so evolutionarily optimized to be robust, the mechanisms are well described, and the BMP determined cells are robust to changes in gene dosage. So when the network is intrinsic to the cell knows how to deal with subtle changes in that network and can buffer that. And molecularly, it's very well characterized how it does. The ventral cells, on the contrary, are not used to BMP, do not know how to deal with it. And oh yes, I forgot to just reinforce that we verify in our developmental reference that this that has been described in Drosophila, in mouse, in model organisms, still holds for the human developing brain. So the two networks are not overlapping, are maintained across developmental points and are maintained across cell types. So we have non-overlapping networks that are intrinsic properties of the cells. So going back to mutational patterns, the mutation that partners with H3.1 in the pons is in 80% of the cases ACVR1. So ACVR1 is constitutively activating BMP in a cell that does not know how to deal with it. Consistently, in the tumors will rely on ACVR1 for having BMP activation. If we remove ACVR1, we lose the downstream phospho-SMAD, we lose the expression of the BMP effectors ID genes, we lose the capacity of colony formation in vitro, we decrease capacity of tumor formation in vivo. So the main point here is that the properties of the cell of origin here are hardwired in the networks of the gene. In this case, it's not necessarily an epigenome conformation like it was in the G34, but a very robust molecular phenotype that will take ACVR1 and BMP as an ectopic signal that produce transformation, and not necessarily in the dorsal BMPs that know how to deal with it. So to recap in this part, what we're showing is that for the tumors we looked at, we are finding very discrete progenitor origins with the G34 in the cortex, and then the different pontine tumors originating in two different ways of OPCs, and the thalamic ones in another OPC way. In these two cases, we could link it mechanistically to why we think the partnering mutations are found in patients. In the rest, there's work to do. So now we have, and the point I want to make is that this is not restricted. This robustness of regulatory networks and properties of the cell of origin is not restricted to the brain tumors. It has been shown in kidney and in melanoma cells, where the actual regulatory networks on the specific anatomical location of the tumors is determining the patterning of mutations, and why we will find RAF mutations in one type of melanoma and not the other. So it's a more general principle than the tumors we're studying. So going back, if I still have time, to the factors we wanted to uncouple. So we're showing that they are originating in different tumor types. The question is now, can we uncouple, not that we know the cell of origin are different, can we uncouple the downstream effect of the mutations, and can we more or less quantify relative effects of the two factors, epigenome of the cell of origin and mutation? So we'll start with this patient that I told you occurs in an epidemioma context, but is driven by H3.1. So when looking at epigenome patterns driven by H3.1, H3.3, and EZIP, we see at the K27ME3 levels very marked differences, as we know and we expected, but we can quantify, for instance, the CGI length, and we see some quantitative subtle differences between the two. At the K27ME2 levels, we see major differences. So we see a very narrow restricted pattern in H3.1, a little bit more spread in H3.3, and a wide spread in ESIP-driven ependymomas. And this can be very easily quantified by the number of PNs that are pasted by these marks. So the question here is, we have a patient that is happening in the context of the PFA. Is the epigenome being reprogrammed to look like an H3.1 when we have an H3.1 mutation? And the answer is, at this level of analysis, no. We see them behaving exactly like the PFA. It doesn't matter if you have ESIP or H3.1. The main component here determining the patterns is the cell of origin landscape. Thus, the H3.1 reprogrammed cell identity for answering this question, cell identity programs in particularly, is it reprogramming the differentiation cascade? So what we did was obtain from Bing Ren's publication single cell chipstick from the OPC and ependymal cells, the two postulated contexts for these tumors, extract genomic features that are specific to one cell type or the other, and then use this feature to cluster our tumors based on acetylation. And ask the question again, what does this patient look like? It looks like a bona fide ependymoma in this clustering analysis, or when we look at bona fide ependymal and OPC genes very well characterized, the epigenome conformation in this tumor for cell identity gene is very much like an ependymoma. So we don't see a major reprogramming of cell identity. And the last experiments we did is in the context of H3.3 and on the K27 gliomas. What happens if we remove the mutation? So for K27ME3, when we knock out the mutation, we remove the allele with the mutation, and this is work with Nada's lab by Ashot and Shakur. We see some spread of K27ME3. And when we swap with the alternative variant, we see again a reversion to the original landscape. So the same CGI lands are the ones that remain pasted with the mark. So we don't see a major effect on PRC2 recruitment. PRC2 lands where it should, based on the pre-programmed cell of origin programs. Same happens with K27ME2. We see a reversion of the patterns, not at the level to K27ME3, but quite similar, and not many differences that we can see genome-wide here. So what would be the cause of the differences that we saw, if you remember, on the spread of K27ME2 mark here? If it's not the actual landscape, original landscape of the cell of origin, the second property, that's a cell intrinsic property that is also very robustly wired in the cellular phenotype that we investigated, is the cycling rate. So the dynamics of the proliferations. H3.1 tumors in general, cell lines, and the tumors proliferate much faster. If we slow down the proliferation by removing ACVR1, and if we make them be more slow, K27ME3, as expected, is not very much disturbed, but now we do see the spread in K27ME2. So again, it's a second cell intrinsic property on the cells that seems to be determining the epigenome patterns we see in the cells. So in conclusion from all those data, what we see is that contrary to what we expected, despite this global epigenome dysregulation, the architecture at these key developmental and patterning genes is very well maintained in tumors. We see it in the G34 tumors, we see in the ESIP and the different histone variants, we see it in the thalamic H3.7ME3. We see that these tumors originate from different ontogenetically defined progenitors, so different anatomical locations would give rise to the different tumors, and those associate with the Histone variant and location and maintain them vulnerabilities. We see that H3.1 seemed to originate in a sonic structural dependent ventral progenitor, and that it relies on ACVR1 for the BMP oncogenic signaling here. On the other hand, these G34 tumors seem to arise in an interneuron lineage and it is the oncogene epigenome state that is specific to this lineage that will favor the co-option of the PGFR oncogene. And so all that I showed you is the result of a very, very close, almost daily collaboration with Nada's lab. So everything we do in these endeavors is done with her. For the questions that will come about CRISPRing in and CRISPRing out and modifying, we are a dry lab, a computational dry lab. So all the analysis is done on our side. And I also want to thank a number of collaborators across the world, particularly the people at the Dana-Farber that provided samples and help. Stephen Mack at NAWA St. Jude that actively was involved in this project, Michael Taylor in Toronto, and then Manat Patani and Paulo Salomon for everything that's related to mouse models that I didn't show but was key for these projects. And with that, I'll take questions.
Thank you very much, Claudia. I will ask everyone to write questions in the chat if you have some. I'm sure there's going to be a lot of questions. I can start with a question maybe about ACVR1. You mentioned about the tumors relying on ACVR1 mutation. So is this gene regulated in some manner? Is it known how this is
it is a transcription factor that is very well involved in brain development. So it is a developmental transcription factor. It's also called ALK, if I'm correct.
Okay.
I don't remember the details of the regulation, the exact cell types where it is. I don't know if you remember, Selin. We looked at it, but I don't remember the details, and I don't want to say anything that is not correct.
That's fine.
Because the details, it's where it matters. But yeah, the effects in the mutations are constitutively activated.
Okay, thank you. I have a question from Afa Al-Bashir. He's saying, great talk, Claudia. Do you think that the defect in spreading of H3K27 dimethyl has to do with catalytic or non-catalytic function of PRC1?
Yeah, so I think it has to do with PRC2, but again, we're looking into this. So these results are quite new about the spread of K27ME2. So I'm not exactly sure about the mechanistic details there. So I think it has to do the differences we see between the histone variants for sure have to do with the amount that we have for each, the cycling rate, and how the mutation would be diluted. So H3.3 is 5% abundant versus H3.1 that is very, very abundant. So if we have a very fast cycling cell, the mutation could be diluted here. And so that would explain the differences in impairment of ME2 disruption, supposed to be one, but further than that, I don't want to speculate too much.
Okay. Thank you, Claudia. I don't see any more questions in the chat. So again, everyone, if you have any question, please don't hesitate to write it down. So if there's no more question, I would like to, oh, there's one, sorry, they come a bit late. Have you looked, again from Afa Al-Bashir, so have you looked at H2K,
No, I haven't.
H2AK 119 ubiquitous?
No,
okay.
For sure, and I know why it sucks, but no, I haven't yet.
Okay. There is another question here. Hello, thank you for your talk from Giacomini Grillia. Hello, thank you for your talk. Do you have any hypothesis on the association of TP53 mutation with H3.3K27M more than H3.1K27M tumors?
So I do, but it's pure speculation. I think H3.1, K27 tumors that appear ventrally in the pons. The reason that appear more often there has to do with the fast replication rate we have in the pons postnatally in the very early years of development of the human pons, where you have a massive glial expansion. So you have to do with the amount of replication that we have there. And the opportunity when you do have this extremely high rate of proliferation in a very short amount of time in this replication-dependent histone. So if that's the case, then it wouldn't need additional P53 help, whereas H3.3 is much less abundant and it happens, throughout. And I think it has to do not necessarily that it needs P53, but those progenitors that are tolerant to both. Is a little bit the survivorship bias. So I think there's something that makes those specific progenitors quite robust to some mutations. So they would tolerate H3.3, K27M, and they would also tolerate TP53. So my speculation is that certain lineage will have more tolerance for some genome levels of genomic instability than others. So that's something we're actually pursuing, but it has to do more to the tolerance of these mutations than in the case of ACVR1, which is actually needed for driving oncogenesis.
Okay, thank you very much.
Speculation with real, of course.
Thank you. I have another question here from Matthew Lawrence.
Yeah.
Great work, Claudia. Is the robustness of K27 trimethyl at native sites driven by PRC1 or PRC2 at such sites?
Again, I think it is K27Me3 with the PRC2 in crosstalk with PRC1, but I don't have the mechanistic experiments here to support any speculation. And in this audience, I don't want to speculate widely on this mechanism. Maybe for a coffee in a non-recorded talk and can widely speculate there. For the second question about the H3.1 PI3K association, yes, we focus here on the three associations we saw, G34, H3.1, ACVR1, H3.3 with non-ACVR1. But actually, the world is very complex here, and we have a number of combinations. And we're looking into every single one of those, trying to make sense of those. So hopefully you will hear about why we think PI3K, but so far we haven't yet figured it out. So okay. Yes, I do think there are some mechanistic associations in each that if we stratify the tumor in the right way and use the right background as comparators, with a very high resolution, we may figure it out. So either itogenic experiments, cell lines that recapitulate the cell of origin, or models that actually recapitulate the cell of origin. There's no point of modeling, I think, H3.1 mutation in a PAX3 neural stem cell because it's not the right context. So, any. So, yes.
Okay. Thank you very much, Claudia. So we're going to stop here with the questions. And I want to again thank you both for these really, really great talks. We were really happy to have you for this month seminar. So thanks again for accepting the invitation. So I would just like, before we end, to remind everyone that we're going to have our next seminar on December 16 with Jimmy Kramer presenting. And I also want to remind all the trainees that we are looking for trainees to join the CERC trainee committee, and the deadline to apply is November 30th. You can look at the website, the CERC website, to have more detail and look into this. So we strongly encourage the trainees to get involved. Thank you very much. Thanks again, Claudia and Selin.
You.
Thanks.